Darwin and Mendel studies have established a new paradigm of species domestication and hereditary laws. More recently, Watson and Crick deciphered double helical structure, and Wennerhaber, member of the Pontifical Academy of Sciences, demonstrated how to restrict DNA, establishing the foundation of recombinant DNA technology. A new era that paved the way for genetic manipulation of important traits among all the kingdom organisms. Currently, results have indicated and support that the assumption that synthetic biology should contribute to intensify studies on the control of gene regulation and design of biological systems. I will make a few remarks and comments on two different items. The first one is the use of recombinant DNA technology and synthetic domestication of useful traits for conservation and add value to biodiversity. For doing that, I'm going to comment on three projects that we do have so far relating the use of the recombinant DNA technology. And the second item is going to be the use of science and technology in Latin America. Having technology as a very important and a key component to progress and reduction of the inequity. I will initiate with the project that we conducted several years ago. We initiated in 2005, trying to mimic the spider silk fibers. It's a partnership between University of Utah and Embrapa. Spider silk has been noted for its physical and mechanical properties. Spiders, you cannot actually cultivate or produce several spiders to isolate the fibers. So, and the fibers are basically protein, biopolymers, and they are forming in repetitive blocks that I'm going to show you in a few minutes. And we did a genome. This is the Latin America map from the Google one. And we collect spiders from the Amazon region, from the Atlantic forest region, and the savanna-like region that we call Cerrado in Brazil. And we conduct the genome for all these spiders. And we were able to develop and to find very primitive spiders with extremely interesting traits that we would like to mimic in the lab. We also add some of this information for a minimum evolution tree for spider proteins that we do have so far. Just as an example, this is a sequence from one of the spiders, which is Nephilensis cruentata. You can note, and the structure of these genes are divided in blocks that are rich in glycine and rich in alanine. You can see that this block is rich in alanine, and we found out that if we create a protein alanine rich, we can actually have a stronger fiber. And you can see if we add alanine, we increase double helix and beta helix in the protein sequence, and we make a more resistant protein. If we use sequences that are rich in glycine, and you can see that there are less alanine here, we are able to develop a more elastic and flexible fiber. And knowing that, we were able to use a natural sequence from the spiders and simulate and create, generate in a computer, the protein to generate a fiber with the mechanical characteristics that we want. So we are able to, as I just said, to control this rich alanine or glycine proteins to generate a spider fiber. And this particular one is one that we were able to, I hope you can see here, 3 times stretching. This is a natural fiber, and this is 3 times stretching fiber. This is still not breaking. So we can use this technology to actually produce fibers for many different purposes. And we do all synthetic in the lab. We produce that in E. coli, in bacteria, because, as I've just said, you cannot actually cultivate and have many spiders. But not only that, that is something that we don't want. We don't want more. We predicted in the future, this is just a model or a new concept for conservation of biodiversity, because we want in the future to find in biodiversity interesting traits and mimic it in a system capable to produce an economic viable system. And then you don't need to go back to the biodiversity to extract more or to collect more organisms or to produce a large scale. I'm not sure if this film is going to, oh yes, thank you. This is a film that we did in the lab, sorry, but I hope you can see the production. You'll see in a bit. We simulate the spirinate from a spider, and using an organic solvent, we can produce as long as we want, 20, 30 meters spider fiber. I hope you were able to see it. So the remarks that I would like to make in this project is that we generate the knowledge to produce synthetic spider fiber and design novel biopolymers. We also show a synthetic domestication of specific traits from the biodiversity and a sustainable model for conservation and add value without doing extractivism. The next project is the production of microbiocides against HIV. This project carries an humanitarian concept that a potential product developed is going to be free of royalties for if produced in developing countries. So this is part of a consortium between the National Institutes of Health in Washington, University of London, Council for Scientific and Industrial Research in South Africa, and Brazilian Agricultural Research Corporation, which is in Brapa. And there was a molecule that was cloned several years ago called cyanovarin, was isolated from the blue gene algae called Nostochys slipsophorum, and is a potent virus inhibitor molecule against HIV, flu, hepatitis, and several others. The problem was that we can produce this molecule in E. coli. However, it's economically not viable because you need to produce a microbiocide, tons of recombinant DNA protein, derived protein, to be able to produce the gel. So we thought to use that in a seed or to accumulate in a seed as a form to reduce costs of large-scale production. Here you can see we introduced this gene in a soybean seed. This is an ultrastructural section of a seed protein, of a seed carrying the black and small molecules here are the oil compounds, and here are the protein storage vacuums that we were able to accumulate this protein in large scale. We were also able to show the recombinant protein derived that we purify it, and the recombinant protein derived bind the GP120 of the HIV virus, comparing production in E. coli and production in soybean seeds. And really is very effective to bind the GP120. We're able to isolate pure, 100% pure protein out of the seeds. And these other pictures show the concentration-dependent HIV activity in soya, and you can see that the range of E. coli that we compare the EC50 values in the range of 0.82 to 0.79 nanomolar in E. coli, and in soybean 0.45 and 1.8 nanomolar. It's very efficient. It is important to mention that the use of seeds to produce pharma compounds is going to be treated as a pharma compound. It's not going to be released in the field and have the potential possibility to interfere in the food chain. This is a care that we should take when we think to use plants to accumulate protein for the pharmaceutical industries. But in the case of soybean, we can produce in a very small greenhouse under control of and not releasing the environment, a considerable amount of recombinant protein, in this case cyanovirine, about 350 micrograms of dry seeds in 18 and less than 250 square meters. So we demonstrated the capacity to accumulate the cyanovirine in transgenic seeds. The remarks that I would like to make out of this project is that we have developed a suitable expression system that we can produce this cyanovirine in large scale. We can also, again, synthetic domesticate a useful trait that was harvested and collected out of the biodiversity. And not only that, we can envisage now to produce an alternative tool for women, and mainly in Africa, to combat AIDS, the AIDS disease, in the case the HIV. Moving now to specific synthetic biology, what we are aiming now is to use synthetic biology to understand essential and non-essential genes in genomes in general. And not only that, to reconstruct partial synthetic chromosomes and understanding how this structure is working. This will help us a lot for we to put genes in tandem and certainly develop products with in a more accelerated manner and at reduced cost. What I'm going to show you now, and I'm going to concentrate, is some of our preliminary results on the use of the integrases to control this specific part. With this, you can see this, the RNA polymerase producing the mRNA, which is inside the nucleus, and is going outside the nucleus and find protein machines, which are the ribosomes, and then it's going to produce the protein that some proteins stay inside the cell and some proteins are secreted. So we've been using this integrase to show some specific way to switch genes on and off. The question was, was serine integrase be able to switch genes in eukaryotic cells that we know that is very efficient and develop regulatory gene sequences. Viruses are DNA or RNA forms extremely abundant on this planet, and most of the viruses are bacteriophages, which can infect and replicate within a bacteria. The process of phage genome integration is mediated by different phage-encoded integrases. There are several ones from different organisms, and even in the same organisms there are several integrases. There are also a very elegant mathematical model trying to explain how these integrases are capable to integrate part of their genomes and excise the genome. This is a figure of bacteriophage. You can the attachment of the bacteria and the attachment of the phage, and you can see that it can integrate according to the necessity to survive, to the phage to survive, can integrate using these attachments sequences within the genome, and not only integrate but excise, and then it's producing two, the attachment left border and the attachment right border. And just to explain how we did develop this regulatory circuits is just as an example, we have, for example, if we put integrase 2 and integrase 5 in an XOR gate, which is going to be, if both integrase 2 and 5 are on, you have no GFP expression. If the integrase 2 or integrase 5 is on, you have expression of GFP. And if you have both either switch on or off, you have 0, as I have just mentioned. So we have the input 1, which is usually a promoter, the integrase 2 and a terminator, and this is important. For example, a dexamethasone external inducer or alcohol external inducer derived promoter that you can actually produce your circuit, and then you can induce switch on and off. What this integrase actually do is, if you generate a construct with both the attachment of the bacteria and the attachment of the phage and the integrase 2, integrase 5, and if you add both, as I've said, both are on, you have no expression of GFP. If one of them is on and the other one is off, you have expression of GFP that you are just going to show you in a few minutes. What happened is, you have here the integrase 2, just an example, and the attachment of the bacteria and the attachment of the phage. And what happened is, the reverse complement is going to do that within the genome and simply switch and flip the DNA, and then you switch the gene. But we can actually, using another integrase, put it back again and make the gene on and off, and we can switch GFP on and off eternally. And the idea is to actually do that with several genes and generate a circuit. Sorry, you probably are not going to be able to see the letters, but these are the product. This is to show that really what we did is, once we've got the results, we isolate DNA and sequence part of what we integrated, just to demonstrate that this sequence out of the sequence was flipped. So it's exactly the same as the attachment R that we add. So it's the reverse complement of the sequence, and the gene is not working, or is on, or is off. And we're able to do that in plant protoplasts. Plant protoplasts, as you probably know, is a cell, is a plant cell without cell wall that we use to facilitate integrating DNA within the cell and further on within the genome. We did also in mammalian bovine fibroblast cells that you can see here expressing, non-expressing, and expressing. And we did also in human cells. And seems that there is a university behavior and good behavior and is working as expected, these integrases from bacteriophages in eukaryotic cells. And we are very pleased with that because we also tested in a plant. This is a tobacco plant because the idea is to actually envisage in the future for example, relating agriculture, we can spray the field with an inducer agent that could be a chemical, physical, or biological to switch specific genes on and off according, for example, the climate. If we have a drought-tolerant plant, we don't need that gene to be on during the whole development, influencing decrease in the physiology and further on in the production. However, we can have a gene because you don't have a drought every year. Some years you don't have drought, so we may be able to switch that on and off according to the necessity, reducing of the productivity or the machinery of the cell to be working it. So the remark on this project is that there are indeed really promising applications of phage integrase in the field of synthetic biology. And the use of integrases for generate nucleic acid parts to assemble pathways and entire chromosomes is really feasible. We can activate and inactivate genes, paving the way for safer gene therapies if we think about humans. And as I've just said, spraying the crops also. So we are now utilizing integrase to determine the endogenous non-essential genes within the different genomes and design and provide systems to control endogenous and exogenous gene regulations, allowing the development, as I've just said, genetic circuits and physical, as I've said, physical, chemical, or biological inducers. The second item that I'd like to quick comment is the item on science and technology in Latin America. Concentrating and emphasizing that technology is one of the key components to progress and reduction of inequity. And to demonstrate that, I'm just going to show you the structure of the agribusiness production in Brazil. This was taken out of the sources, the agriculture census, that the data that I've used it for. In Brazil, we do have about 5 million farms, establishments, about 5 million. And 3.3 million represents 64% of the total, and 1 million represent 30%. But if you see the production, the gross value of production, 3.3 million produce only about 20%, 1.6 million produce 80%. What's the problem of that? The problem is that we still keep more than 50 million people in the poverty over the last decades. And why is that? Have a look. We divide that in 3 segments. One that can make more than $25 a year, the other one between 5 and 24,000 a year, and the segment D and E that they can make $5,000 a year. So are very poor farmers. But you can imagine they are occupying about 100 million hectares. If you put together with not very small but technified farmers, you can have the same area that we are using with a very low productivity. So sorry for the long thirst, but I think I'd like to remark and to emphasize what I'm just going to say, but producers are surrounded by a social and economic environment that discriminates against the low-income class segment. Low-income farmers commercialize production, they output at lower prices and purchase inputs at higher prices. Therefore, utilization of technology implies an inadequate ratio of inputs at a higher cost to generate an output. That's not profitable. In contrast, and as a consequence, the technology is barely adopted. In contrast, without technology, there is no way to increase the revenue per hectare and open the door to alleviate poverty in agriculture. That's the truth, because we realize with the census data that if we add technology within the systems, is the only form for them to jump to the other segment and go to the segment that makes about $5,000 to $24,000 a year. So, okay, it is only a compilation of the obvious. There is nothing new. And indeed, there is nothing new, but it's words for which you mentioned that, in my view, only the equalization components will be the key for which to get progressive development. Developed countries were able to build an equalized foundation to sustainable progress based on science and technology. In contrast, most of developing countries were unable to establish an equalized operational foundation, deriving an ability for a sustainable progressive development, including reduction of inequity. So, in my view, and to put short, and being reductionist, we need to set an equation. We need education, science and technology, human health, environment, adding value to have the progress. It's not only technology. Technology is a very important component, and even a decisive component for which to jump, but it's not enough. Trying to apply technology without all the other components, you are not going to succeed. So the remarks on that.
Excuse me, Professor Reck, we need to leave some time for discussion.
Okay, so I'm just finished. Just three seconds more. This is the last slide. The remarks is what I've just said, that the key is the key component, but no without all the other components. Thank you all.